Armaroli from Newark, who will present her work on elephant ecology in a Neanderthal landscape. Elena, you authorize us to record the talk and then upload it on the PAASTA YouTube channel. So we will start the recording and then you can go ahead with your presentation.
Okay, thank you very much. First of all, for the invitation. So I'm very happy to be here with you today. I am a postdoc in isotope analysis. Actually, I'm in Spain under an ERC project in the Basque Country. Actually, I'm in Lleida because I'm carrying out some isotope analysis here. But yeah, this work was part of my PhD when I was a student in Modena, at the University of Modena in Italy. And I will talk about elephant ecology in a Neanderthal landscape, so the landscape of Neumark-Nord, which is a site located in northeastern Germany, dated back to the last interglacial. This work involved multi-proxy. It was a multi-proxy work involving both polyproteomics and multi-isotope analysis. Unfortunately for you, I guess I will not spend many words on polyproteomics because I am not an expert on that. I collaborated to the analysis, but my research field is in isotope analysis, so I will focus the presentation on the results of the isotope analysis. So first of all, what kind of material we targeted? We used the tooth enamel because as all of you probably know, it is made up by approximately 95% of hydroxylapatite. So it is very, very resistant to postmortem modifications thanks to its crystalline structure. Also, it grows incrementally in years, depending on the species. So from a few years to 8 to 10 years, as in case of the elephants. And after the end of formation, it doesn't remodel anymore. So it retains many information on the first years of life of the individuals. So it is a real biological archive for the study of animal behavior in the past. We performed on the enamel both amylogen analysis for sex estimation. This was the most innovative analysis that we performed because it was the first time that this kind of analysis was applied on Paleoloxodon antiquus. And also we applied multi-isotope analysis, mainly carbon, oxygen, and strontium isotope analysis. I highlighted these elements in the general formula of the hydroxylapatite. So carbon and oxygen are present in the carbonate group, substituting for the phosphate group of the hydroxylapatite. So we measure the carbonate portion of the apatite. And this general formula is made more complex by a number of trace elements, including strontium. Strontium has a similar chemical behavior to that of calcium because they belong to the same group of the periodic table. So strontium substitutes for calcium in small amount in our apatite. So we measure the radiogenic strontium for the study of mobility. To sample the tooth, different methods can be used. The first one is bulk sampling. I do not write it here, but it means simply drilling the whole surface of the tooth to obtain a mean value of the time period represented by the tooth. But in this case, we wanted to reach a higher spatial resolution. So we sample the teeth both sequentially for carbon and oxygen isotopes. And this means sampling the tooth from the apex, where there is the oldest enamel, to the enamel root junction, where there is the younger enamel, sorry, like you see in the image in the drawing on the left. Also, we applied in situ isotope analysis for strontium, which means working with laser. As you see in the image on the bottom of the slide, but I will show you this in greater detail later on. With this technique, we can reach a sub-seasonal resolution. So it is very precise. What are the principles behind the use of isotope analysis in archaeology? Oxygen isotopes varies geographically according to different environmental parameters such as temperatures, latitude and latitude, humidity, precipitation, etc. So varies geographically. That's why it is widely used in provenance studies, very often together with strontium. But also, it is widely used for paleoclimatic studies. So, for example, for the reconstruction of the temperatures. On the other hand, carbon isotopes varies according to the photosynthetic pathway used by plants, because carbon is the carbon contained in plants that animals eat. So this value varies according to the photosynthetic pathway, allowing to distinguish between C3 and C4 plants, of course, in the environment in which both these plants are present, because C4 plants are typical of tropical and subtropical regions. So, for example, in temperate regions such as Europe, they are almost absent. But in C3 plant ecosystem, it's still possible to make some inferences on the environment, as we will see later on. Carbon isotopes are very useful for reconstruct animal diet and also for the environment and habitats in general in which this animal lived. Strontium, we use this ratio, which is the ratio between the strontium-87, which is the product of the radioactive decay of rubidium-87. This decay takes very long period of time, so we can consider this ratio stable, more or less, for our period of interest, so in archaeology in general. But it varies geographically depending among the different lithologies present in the environment. So according to the age of rocks and their initial isotopic compositions, it is possible to trace the provenance of the individuals, because strontium basically passes from bedrock to soil through weathering processes mainly, and then from soil it enters water courses, seas, etc., and also is taken up by plants through root uptake and entering in the end the food chain. So overall, carbon, oxygen, and strontium are taken up through diet mainly. And strontium provides many information about the mobility and also the provenance of the individuals, thanks to the use of isoscapes like this one, which is the isoscape of Europe, showing in red the more radiogenic areas, and in blue the less radiogenic areas. And maps like this one are widely present today in the literature, so they are available for many parts of the world today, and they are very useful to track the provenance of animals. So how do we work in the lab? Strontium can be separated through chromatographic separation in a clean room, for example. So in this way we work in a solution mode, meaning that the enamel chunks need to be separated from the matrix. The first step is the digestion of the sample through a weak acid, mainly nitrogen, nitric acid, sorry. And then the sample is separated through a specific resin. So strontium is separated, and then it is measured by mass spectrometry. In this case, usually we use a multi-collector mass spectrometers, which allows to measure multiple masses simultaneously. On the other hand, we can work on with laser, which means that enamel slabs, as I will show you later on, are directly put into the laser ablation system, and then the enamel is sampled with a laser that take a very small amount of sample, sort of aerosol of the sample. So which means that we have a minimal sample loss with this technique and a higher resolution. And then the laser ablation system is coupled with a mass spectrometer for the actual measurement of the isotopes. For oxygen and carbon, we need to use another mass spectrometer, which is the isotope ratio mass spectrometer. And in this case, the samples taken from the teeth are powders that are directly put into the mass spectrometer, and they are acidified by an acid, orthophosphoric acid, which creates which liberate the CO2 of the sample. So basically, we measure the CO2 of the samples, so meaning that both carbon and oxygen are measured simultaneously with this technique. But why we targeted Paleoxodon antiquus? Mainly because, first of all, there is not much literature on this species, because other species such as mammoth are more studied, as in this case. This very nice work from Kowalik and colleague published in 2023. In which they unraveled the ecology of the mammoth, measuring strontium in situ, coupled with oxygen isotopes measured sequentially, as we did. And they found evidence for migration of mammoth. And we would like to get similar results for our Paleoxodon antiquus. Because in literature there are few information on the ecology of the species. A recent work published by Roditi and colleagues found do not find found evidence of migration. But yeah, these are more ancient samples and in Greece. So we would like to know more on the Paleoxodon antiquus for more recent time, and specifically Paleoxodon antiquus found at the site of Neumark-Nord, which is located in northeastern Germany, in Saxony-Anhalt. Today, the site is not visible anymore, unfortunately, because it's covered by this artificial lake. But in the past, many decades or excavations found evidence of a Neanderthal presence at this site. Here there was a mine, so the rescue excavations go ahead during active mining of this place. And they involved year-round archaeological excavations and many international students working year-round under the supervision of the University of Leiden and also the Morepos Archaeological Research Center. So today, so the site is not visible anymore, but in the past, it should be something like this. This is a very good image provided by my colleagues, showing how the landscape of Neumark looked like during the last interglacial. So it was basically a lake-dotted landscape. So it was characterized by the presence of many lake basins. And the archaeologists found evidence for two main sites named Neumark-Nord 1 and 2. It was about 30 hectares of lake landscape in a semi-open environment with excellent preservation of organic remains thanks to the presence of water. So they found many flint tools, for example, and also evidence for fire use. In fact, Neanderthal helped keeping this landscape open through the use of fire, meaning that they were able to live in a forested environment. And also, they were present here almost continuously for more than 2,000 years, meaning that they were more sedentary than previously thought. Among the faunal species whose remains were present in a great amount. Are present among the fauna species are present both carnivores such as hyenas, lions, etc., but also many herbivores such as cervids and horses, for example, and also elephants. Elephants, which are forest species, because Paraxoranticus is a forest elephant, and here more than 70 individuals were found, and this is the greatest assemblage of Paraxoranticus found so far. Paraxoranticus, from a genetical point of view, is a close relative of extant African elephants. And it was characterized by pronounced sexual dimorphism, with males reaching up 13 metric tons and 4 meters only at shoulder. So they were massive, and they were the largest terrestrial mammals of the entire Pleistocene. Very interestingly, most of the skeletal districts display anthropogenic cut marks on fresh carcasses. And this evidence, together with the unbalanced mortality profile skewed toward adult males, means that Neanderthal actively hunted, so targeted specific member of the group, elephant groups and populations, to get meat, first of all, but also marrow, because as you can read on a recent paper published in 2025 by my colleagues, in a specific part of Neumark-Nord 2 sites, that there was the so-called fat factory, in which Neanderthals actively extract marrow from bones, which is a very time-consuming and a very long process, and complicated also. And this kind of process is visible in later times only in Upper Paleolithic, but Neanderthals practiced the marrow extraction at Neumark. And also, the archaeologists demonstrated that, calculated that for a group of 25 people, from 3 to 5 days were necessary to complete the whole process, so from hunting to processing and marrow extraction, etc., providing meat for months, so at least 3 months. So it was necessary for them to store this meat in a way. So probably they knew a way to store meat, so with techniques such as smoking, drying, etc. So overall, Neumark-Nord is an impressive window on Neanderthal behavior during the last interglacial. So our analysis, starting with this part, finally, we selected, overall 4 molars pertaining to 4 different individuals. Before the starting of the project, a colleague performed bulk sampling on these teeth, which show that some of the elephants displayed strontium values compatible with that of the site, and others don't. So we selected 2 local and 2 non-local elephants for the analysis, which involved both amylogeny analysis, as I told you, for sex estimation, in situ laser ablation strontium isotope analysis to assess the movement behavior at a subseasonal scale. We also built an ad hoc strontium isoscape of northeastern Germany for provenance assessment. And finally, we selected 2 teeth for carbon and oxygen isotope analysis to study the habitat of these elephants, their diet, and also find evidence for environmental seasonality. Starting with amylogeny analysis, amylogeny was extracted through routine method in a clean room, making use of stage tips, and then it was measured by tandem mass spectrometry. Overall, we found 3 males in our data set, and one possible female. This individual was labeled possible female because of the lack of unique amyloid peptides. So, starting with strontium, for strontium isotope analysis and for isotope analysis in general, the teeth, which were very, very big and very, very heavy, we need first of all to cut them using very big saw. After cutting, we selected the longest lamella, which grows, all the lamella grow simultaneously. So simply we selected the longest one, which represented the longest time span, and which was more or less 8 to 10 years. And also the macroscopically best preserved in order to avoid contamination, a possible contamination. Then these labs were put into the laser ablation system, as you see in the image on the center of the slide, together with some standards to correct the data. So before starting, we need to draw the laser path, and we draw this laser path at the enamel dentin junction, because here there is the best preservation of the isotope signal, because there is not yet the overprinting of from successive enamel layer deposition. Here there is a cute GIF of the laser working, and the spot size is of the laser is very, very small, more or less 100 micrometers, and the scan speed was more or less 1.5 millimeters per second per second, sorry. So considering that the slab were more or less 15 centimeters, this kind of analysis requires a lot of times. And also we ran few lines of dentin just to be sure that we were measuring the biogenic strontium, so not contaminated by the soil, for example. Here the results. As you can see in the first panel, the two couples of elephants display different strontium values. So there are two elephants that have less radiogenic values and two elephants that display more radiogenic values. The two displaying less radiogenic values correspond to the local baseline, which was assessed by bulk measurements of teeth of other species found at the site, which are also represented by the box plots. But overall, all the elephants display a high degree of mobility, as you see from the very variable profiles. But the most interesting thing is that these two couples of elephants seem to not have roamed in the same geological area for the 8 to 10 years represented by the teeth. Oh, and also the diamond are the dentin measurements, and as you see, all the dentin samples are compatible with the local strontium values. So, which means that we were measuring the biogenic signal overall, and also possibly some biogenic strontium was still present in dentin because the orange and purple diamond, which belong to the most radiogenic individuals, have higher values compared to the other two. And then, as I told you, we built this small isoscape of the area, making use of literature data mainly. So we used modern water values and other archaeological strontium values published in literature to build this isoscape. And yes, the white dot is the site of Neumark, and for that we use the baseline values. And as you see, there are some radiogenic areas around the site, while the site itself has less radiogenic values. And then through a Bayesian approach, we estimated the origin of the elephants. Here is just the workflow to show you how it works. So the potentially local individual, as you see, have a value corresponding to the provenance correspond to the site of Neumark. So the yellow areas, which are the most probable area of origin, correspond to the site, while in the other case, the highlighted areas are far away from the site. So here in a greater detail, what we did for all the elephants, we estimated the density. So on the left, you see the density plot with the dashed lines representing the modal values, so the values most present in the profiles. And overall, for all the elephants, we estimated the provenance for the all modal values and also for the maximum and minimum values. So that's why you see so many maps in the center of the slide. But in the end, we merged all these maps for every elephant to get the final probability maps of origin that you see on the right. This map shows the top 5 provenance probability. And as you see, the green and the pink maps of the local individuals have highlighted the highlighted areas correspond to the site. While on the other hand, for the purple and orange individuals, the highlighted areas correspond to other areas far away from the site. And this site, as I will show you again the map, these areas are the red one here, and geographically they correspond to mountain ranges that are around Neumark. So going ahead with carbon and oxygen isotope analysis, for this analysis, we selected only 2 teeth. The one non-local male and one possible female, which has local strontium values, to see if there were differences between male and female, but also to see if there were differences in terms of diet and habitat for the local and non-local individuals. I performed a micro drilling using a very small tip, but of course it was not possible to reach the same spatial resolution of the strontium isotope analysis, but I followed the laser line for the sampling, and I was able to obtain more or less 70 enamel samples for one tooth and more than 90 for the other. Here are the results. As you can see, both carbon and oxygen isotope profiles are quite variable, which is in line what we expected for the last interglacial. So the climate was temperate and marked by environmental seasonality. Intratooth oxygen isotope values reflect our continental climate. And also, on the other hand, the carbon isotopes are not very variable, but because we are in a C3 plant environment, so the values don't change very much. But the small variations that we can see allow us to say that the elephants have a plastic behavior in terms of diet, meaning that they eat whatever they found in the environment depending on the season. So as we know from the comparison with modern elephants that are very plastic also in this sense, so they eat barks, leaves, fruits, whatever the environment has to offer. Unfortunately, we were not able to find a direct comparison match with the strontium isotope profiles. So we were not able to say if these elephants were migrants or not. In part because of the differences in sampling in the sampling strategies, but also in a way this is in line with what we know from extant species, because we know that elephants in some populations migrate, but others do not. And there are also differences in the same individual. So one year, one individual can migrate and the next year not. It depends on the local climate and many other factors. So we found many comparisons with extant species overall. The most interesting result was when comparing the carbon and oxygen isotope alone, because as you can see from the box in the center, these individuals form completely different clusters. With the more endogenic individuals displaying more negative values, both in terms of oxygen and carbon isotopes, which means that this individual probably roamed in a more forested environment, because more negative values usually indicate forested environments. And also in terms of oxygen isotope, this means that this animal drank from distinct water sources characterized by more negative values of oxygen, so more cold. The environment in general was more cold, more humid, and also characterized by different elevation, probably. So all these elements overall are in agreement with the possible provenance from the mountain ranges around Neumark. So to conclude, we were able to assess for the first time the sex of Hyloxodon antiquus through amelogenin analysis, and we found evidence for Neanderthals that were able to actively hunt both males and female individuals of straight-tusked elephants, which again were the largest land mammals of the entire Pleistocene. And through multi-isotope analysis, we were able to found evidence that there were probably two distinct populations at Neumark, as we saw from distinct isotope values, both in terms of strontium and carbon and oxygen, meaning that they roamed in distinct environments in terms of geology, vegetation, and water sources. We also confirmed that the landscape around Neumark was semi-open, and probably this site acted as a point of attraction for animal species, so such as elephants, for example, but other animals that were found at the site. And this probably make Neanderthal use this site almost continuously for more than 2,000 years. So overall, this multiproxy approach provides a powerful toolbox to get many information and unprecedented insights into the ecology of straight-tusked elephant, and it is possible to use this kind of analysis to unravel the ecology of extinct species. And last but not least, I would like to mention that it's also possible to make a comparison with extant species. This is an image of Zanga Bay in Central African Republic. In Central Africa, today are present these clearings in the forest, known locally as bay, which are characterized by the presence of water, mineral sources, and many plant species. And these sites are aggregation sites in which elephants meet to socialize and also find mating opportunities, etc. So they periodically meet here, while the rest of the year they roam in the canopy environmental, under the canopy of the forest, and many are very solitary normally, but they meet at this meeting point during the year, and actually Neumark looks like this kind of forest clearings. This work was recently published on Science Advances, so if you are curious and you want to know more on straight-tusked elephants at Neumark, you can read this paper, and here is the title. And to conclude, I would like to thank all my colleagues. Unfortunately, I don't have pictures of all of them, but I am very grateful. I had a lot of fun working on elephants in Frankfurt and also in Mainz, where the analyses were carried out. So I thank all of them, and of course, all of you for your attention.
Thank you, Elena. Thank you for this great talk. Um, so we have some time for questions. Um, I guess you can just raise your hand or ask anything in the chat and we'll read it. I can start with a question. Can you hear me, Elena? Yeah.
Yes, yes. I cannot see you, but I can hear you.
Oh, maybe you can stop sharing your screen and you'll
Yes.
see us.
Okay. Okay. Now, so. Yes.
Yeah. So I think, yeah, this is a really great example of a multidisciplinary, multi-proxy approach that's really interesting. I was just wondering, having both the proteomic aspect and isotopic analysis was planned from the beginning or not? And if so, how did that affect maybe the sampling strategy of the tooth? Like, did it have any influence on where you would sample and in a way to minimize the sampling of the teeth?
Okay, yes. No, I think that the proteomics was not in the program at the beginning because, yeah, we would like to understand more on the ecology of the elephants. But since, as I told you, we saw at the site we found evidence that Neanderthals found more males than females. In the end, we decided to try because it was a first try because it was the first time that this kind of analysis were performed. So yes, we basically after that after the sampling for isotope analysis, we just relevate a small amount of sample for amylogenin analysis, but from the same individual from the same slab. And yeah, and in the end we got the results, so we decided to publish again with the isotope analysis because it was very exciting to have this analysis done.
And do you think they would do more maybe of these analysis? Because yes, you potentially have a female, but you also have three additional males. So that could actually confirm that they were mostly targeting male elephant. But do you
Yeah,
know if there's plan to do more of sexing analysis?
yeah, this is a good question. I would like to perform more analysis because yeah, it would be useful to have um, an overall view of the herds present at the site. But yeah, these analyses are very time consuming. So and now I think that they are not in program. But maybe for a future project with extra funding, maybe yes, I would like it so much. Yes.
Okay, thank you. Do we have any other question? Okay.
Okay.
Well,
I have a
anyway, this talk
question.
will be.
Well, it's not related to the strontium analysis, but much about the site. Like, I'm curious if in the site there's also some um, bone tools or other things made with this bone species, like or like a I don't know musical instrument like a flute or something. I don't know. I'm just curious if you find something different that is not just the remains from the hunting.
Okay, I think no, no instruments in terms of musical instruments, but maybe some object useful to work materials, etc. I think maybe, but I'm not sure. I don't remember for sure. There are many flint tools, but bone tools, I'm not sure. I think that they were able to create them because they were able to do many things with the bones. So why not tools? But yeah, I'm not sure. I will need to check the other papers published on the site.
Okay. And another question also just because I'm curious about this. Are you planning to do more paleoproteomic analysis with these individuals that you have? Or is something that someone else is going to carry out?
I would like it so much, yes. Also because I would like to learn more about paleoproteomics because I collaborated in the extraction of the amelogenin in the lab. But yeah, I would like to learn more about the bioinformatic analysis. So I think that if I will write a project in the future, I will include the amelogenin analysis. But yeah, for the moment, they are not in program, unfortunately. But I know that DNA analysis are in course, are underway. So yeah, they will confirm. I think the evidence we found with amelogenin.
That's very cool. Thank you for your presentation.
You're welcome.
I give a few more minutes in case there are any more questions. But again, yeah, your talk will be uploaded on the PAASTA YouTube channel. So for anyone interested that could not attend this event, they can rewatch it. Yeah, if there are no more questions, I think we can finish this talk session. Thank you very much again, Elena, for doing this talk. That was really interesting. It's such a cool site.
Yeah.
Thank you
for
very
the
much.
invitation.
Yeah.
Thank you, Elena.
Okay.
Bye.
Bye.
Bye, bye. Thank you.